Hello dear students, I'm Dr. Moin. Students, as you know that we have gone through various techniques involving in thermal methods of analysis. So we have discussed thermogravimetry, differential thermal analysis, differential scanning calorimetry, evolved gas analysis, etc. But in current video, we are going to discuss about differential photocalorimetry, DPC. So let's get started. Before starting the video, if you didn't subscribe my channel yet, then subscribe it right now and also press the bell icon so you may get in touch with my upcoming videos. And here are the contents of current video. So in this video we'll see the basic principle of differential photocalorimetry, then we'll see the introduction of the technique, then we'll see the complete instrumentation, a detailed instrumentation of differential photocalorimetry with the help of various schematic diagrams, and then in the end we'll see the applications of DPC. So first of all, principle of DPC. The basic principle of differential photocalorimetry involves the interaction of light into the reference and sample cells of calorimeter. So this technique is quite very much similar to that of DSC, but the difference here is the light, DPC. Photo means light. So here there is the interaction of light into reference and sample cells of calorimeter. If a photoreaction occurs, the signal in the sample side of the system will be out of balance, and light interact with both the cells, that is reference and sample cells. And if there occurs some sort of reaction due to this light, then definitely the signal in sample side will be out of balance as compared to that of the reference side, resulting in a thermal output which is observed by the calorimeter, and this difference in signal is observed by the use of calorimeter. So this is the principle of DPC. Let's see the introduction. Photocalorimetry is considered as an extension of classical calorimetry. So it is just the extension of the simple calorimetry. And what is the purpose of this technique? The determination of enthalpy or internal energy, which is also done by use of that traditional calorimetry. So determination of enthalpy or internal energy changes associated to radiation-induced process. So the difference here is only that these processes are being started here by the use of some radiation. Many different kinds of photocalorimeters have been built and used, and these generally include, so the main parts of these photocalorimeters are a standard calorimeter, so definitely it includes a standard calorimeter, then an optical irradiation system through which the radiations will be generated and made to fall on the reference and sample cells, and then a connection between both of these two parts. So these photocalorimeters are useful for some specific type of reactions. Let's see. So these are particularly useful to study reactions which are too slow for observation under thermal activation conditions, but that can be efficiently activated by radiations. Means those reactions which are too much slow under the action of heat, and they are quite easily activated by the use of some sort of radiations. So such reactions can easily be studied by the use of photocalorimeters. They also find applications in the study of complex photochemical processes for which a complete thermodynamic analysis based on literature data is virtually impossible. So there are various photochemical processes, means those reactions in which the light undergoes some sort of chemical changes. So these are also being studied by the use of these photocalorimeters. The photocalorimeter provides a way of monitoring such processes, like yielding information about the heat being absorbed or evolved, the kinetics, the quantum yields, etc. So by the use of photocalorimeter, we can gain various type of information, like whether the heat is being evolved or absorbed, means whether the reaction is endothermic or exothermic. We can study various types of kinetics of reactions. Then quantum yields can be studied by the use of these photocalorimeters. Examples of these processes are photosynthesis, chemical reactions associated with the chemistry of vision, then photopolymerization, polymerization in presence of light, and photodegradation, the breakdown in presence of some light. So all these processes can be well studied with the help of photocalorimeter. DPC is a new and powerful analytical tool. So it's a totally new technique which is used for characterization of ultraviolet curable materials. So such materials can also be studied by the use of this technique. DPC is an adaptation of differential scanning calorimetry, DSC, which involves the use of an ultraviolet or visible light source to study the behavior of photoreactive systems and have specific applications. So just I mentioned earlier that it is just like differential scanning calorimetry. The difference is only that the reaction is started over here by the use of some light, and the light may be ultraviolet or visible light, and in this way the behavior of various photoreactive systems are observed. Differential photocalorimeter involves shining light, as I mentioned earlier, maybe ultraviolet or visible light, and this is made to fall on reference and sample sides of the calorimeter. And if a photoreaction occurs, the signal in the sample side of the system will be out of balance with respect to that of reference side, and thermal output results, which is observed by the calorimeter. So the light is made to fall on both sides, that is reference and sample. So if the reaction starts by this radiation in the sample cell, then definitely the signal from sample side will be out of balance, and this out of balance is measured with the help of calorimeter. In general, a photo-induced reaction requires the determination of two quantities. So we determine actually through these reactions the two quantities. Let's see which are these. The change in, first one is the change in energy of the samples brought about by isothermal photochemical transformation. So what is the change in energy of the sample which are brought about by these photochemical transformation? So first thing is to measure the change in energy, and second thing is the amount of materials in moles transformed in the reaction. So how much quantity of that material, and that is in moles, which is transformed due to this photoreaction. So these two quantities, actually these are determined with the help of a photo-induced reaction. The next one is instrumentation of differential photocalorimetry. The basic components of a typical differential photocalorimeter are light delivery system. So definitely we need a lamp source for the production of ultraviolet, for the production of visible light, and power supply system for the generation of radiations, so to make these lamp on. Then there will be the filters or monochromators, so if we need some monochromatic light of single wavelength, and then appropriate optics for beam steering. So there are some optics for beam acceleration. So this is all about the light delivery system. So this will be present in the instrument. The next part will be the calorimeter, which contains the sample and provides the actual measurement. And the last part is our recording device, which also contains a personal computer. And here is the schematic diagram of a photocalorimeter. So we can see here is the lamp, which may be either of ultraviolet or visible light. So light will be generated from here. And next one is the optical system. We can say the monochromator system. There may be entrance slit, exit slit. So monochromatic light is then made to fall over through this mirror to the next part, which is the calorimeter. And calorimeter, actually it contains the both reference and sample holder. So light is made to fall on both the cells. So photoreaction start over there, and the signal from the sample is out of balance. So these signals are then made to fall on next system, that is amplifier and analog to digital converter. Because the strength of the both the signal is different, so alternating current is generated over here, which is amplified on amplifier, and this so current is generated over here, and this signal is fed to the recorder pen, which make the graph. We get the peaks on the screen of computer, and this thermogram can be printed by the use of printer. So these are basic components of the instrument used in DPC. Then there is another diagram, and this is the schematic diagram of various internal components of photocalorimeter. So internal components, let's see. So these are pointed here by 1, 2, 3, 4. Let's see what these show actually. So here is one, and what does it show? It is furnace surrounding. So this one is the furnace surrounding. Okay. So number 2, here is the number 2, and this is the heat conduction path in the form of radiation. So this one is the number 2, heat conduction path, or radiations are being moved from here to these different sides from sample and reference. So this is the conduction path of these radiations. Then number 3, you can see over here, this is the number 3, and this shows the sample support. This is the sample support. Then this is the number 4, and this shows the sample container. So it is the container, and it contains the sample, which is shown by here with the number 5. So this is sample. Then number 6 here, this is the reference material, and number 7 is the this is the container which is containing the reference. So this is called reference container, and this is number 8, which is called reference support. So this is all about the different parts, internal components of these photocalorimeters. Then there are different signs which are shown over here, and these will be used in the coming slides. So we must be known what these are actually. So for example, this TF, we can see here is the TF. This is the called temperature of furnace. So TF denotes the furnace temperature. Then TF initial, so initial temperature of the furnace. So beta could be the another sign which represents the heating rate. Small t, this is the time. Then TS, which is shown over here, TS. So this is the temperature of the sample. Then TR, this is the temperature of reference. Then a sign used over here, phi. Phi is the sign for heat flow or heat flux. So phi Fs mean heat flow from furnace to sample. So this one, phi Fs, you can see over here. This represents the heat flow from furnace to sample. Then phi Fr, and this shows the heat flow from furnace to reference. Then another sign is the phi SR, and that is the heat flow between sample and reference. And let me tell you that the heat here is generated by the use of these radiations. So these must be kept in your mind. In the ideal steady state, so in the ideal condition, the sample and reference substances have the same temperature. So the temperature of both sample and reference substances is same. And the temperature of both of these is lower than that of furnace temperature. So we can write it like this, that the sample temperature is equal to reference temperature. And the temperature of both these is lesser than furnace temperature. The difference, and what the difference? Furnace temperature, the difference between furnace temperature and sample temperature, actually it depends on the heating rate, the thermal conductivity, the heat transfer coefficients of the surfaces, and the heat capacity of the sample. So the difference between furnace temperature and sample temperature, these depend of all these factors. So this difference will be lesser if the heating rate is higher, thermal conductivity is higher, if there is high heat transfer coefficient of the surfaces, and same is the case with the heat capacity of the sample. So the difference between furnace temperature and sample temperature will be lesser. Then every release of heat in the sample reduces the temperature difference between surrounding and sample. So definitely more the heat will be delivered in the sample, so it reduces the difference between the furnace temperature and sample temperature. That is, heat transfer from furnace to sample becomes smaller, or even it changes its sign if sample temperature is greater than furnace temperature. So might be there could be the possibility that the temperature of sample could be high, so there will be the change in the sign as well. Then in case of an existing temperature difference, a part of heat flow generated in the sample flows along possibly existing paths to the reference substance because of the temperature difference between sample and reference, which is a measure for this portion and consequently for the heat flow of the reaction as well. So now the heats are flown towards sample and reference pan, and there is some reaction which has occurred in the sample pan. So there might be possibility that there could be the temperature difference between both the sample and reference pan. So heat could flow from the sample pan towards the reference side, and which is measured, and which gives actually the result. Thus the heat flow due to the reaction in the sample is proportional to the difference between heat flow from furnace to sample and heat flow from furnace to reference. So how much heat will be flown from the sample to reference? So it is actually proportional to the difference between the heat flown from furnace to sample to furnace to reference. In other words, the difference in temperature between sample and reference measures only the difference between the two heat flow rates, towards sample and towards reference side. As a consequence, fluctuations of the temperature of the surroundings which in the first approximation have the same effect on heat flow from furnace to sample and heat flow from furnace to reference. So fluctuation of the temperature of the surrounding, these have the same effect towards the heat flow from furnace to sample and furnace to reference. So actually this don't influence the difference between these two heat flows, so that the temperature difference between sample and reference is not affected by the change in temperature of surrounding. So this is one of the essential advantages of the differential principle, while another advantage is the better signal to noise ratio when measuring the small changes of a large signal. So these are two advantages of this principle. And in the end, the applications of differential photocalorimetry. Calorimetry is a primary technique for measuring the thermal properties of materials to establish a connection between temperature and specific physical properties of substances. So this is the basic definition of calorimeter, or its function, the function of calorimetry. It is the only method for direct measurement of enthalpy associated with the process of interest. So any kind of heat change that can be measured by the use of this calorimetry. Calorimeters are being used frequently in various fields like chemistry, biochemistry, cell biology, biotechnology, pharmacology, and recently in nanoscience to measure thermodynamic properties of biomolecules and nano-sized materials. So here are different fields and different processes in which DPC is frequently being used. So in biology, it is being used in various processes like photosynthesis, photomorphogenesis, phototropism, studies on plant tissues, then metabolism of living cells and microorganisms, then in study of luminescent bacteria, yeast, and viruses. So in all these studies, DPC is being used. Then in chemistry, it is being used to study the fluorescence of organic dyes, determination of quantum yields, solar energy storage in organic chemistry, like different compounds like coordination chemistry, including organometallic chemistry. Then in material sciences, DPC is being used in the processes like photodegradation, photopolymerization, etc. So dear students, this was all about current video, but still there are a large number of videos in the pipeline. So to get them in touch with all of those videos, you need to subscribe my channel. So thanks for watching. Thank you very much.